So first of all, biomolecules means what? So bio means living and molecules mean certain small components which are present mostly where in the living organisms. So biomolecules means what? It's nothing but living or chemical molecules present in living organisms. So basically these are of two types. One which comes under the organic and the other one is the inorganic. So organic and inorganic means under the organic, you will be having the simple larger proteins like proteins, okay, carbohydrates. Then we can take vitamins, nucleic acid, and so on. So the bigger components. Then in the inorganic, it will be mostly smaller ones like minerals, water, or even gases we can take. Okay, so in this chapter, we'll be learning about the carbohydrates, proteins, and all those like macromolecules, okay, in detail. So, like how it is categorized into organic and inorganic, in the same way, it is again categorized on the basis of its structure, means how big the size is, into two types, that is micro and macro. Micro and macro molecule. So again, the same thing, only the thing is the term will be different. So micromolecules mean small molecules they will be. So it will include all the, what do you say, the short components which is going to make up that particular big component. For example, we can take sugars or we can take amino acid. Okay, so sugars means when the sugar molecules come together, they will form something big, which is nothing but the carbohydrates. Or if you take amino acids, so they are going to form what? Proteins. Okay, so these are what the categories of the biomolecules depending upon the origin as well as the size of that particular component. So first and foremost, we are going to see about the carbohydrates. Now we all know carbohydrates are nothing but the components which provide us with the source of energy. So only if we take the carbohydrates, there will be a lot of energy and all the biochemical reactions will take place in a cell. So carbohydrates basically are sugar or saccharides. Okay, so saccharides meaning only sugar. So they are particularly made up of what? The sugar molecules. Okay, so what are the properties of these carbohydrates? So the first thing is what? Carbohydrates means they are optically active polyhydroxy aldehydes or ketones. Okay, so CHO, C double bond O. Aldehydes or ketones. Okay, so depending upon whether it's having a aldehydic group or ketonic group, okay, there are different types. Then depending upon how it's going to react with a particular, what do you say, chemical, whenever we do a test or whenever we take a carbohydrate and treat it with some chemicals, what test is it going to give? So depending upon all those factors only, we are going to see the types as well as the classification of the carbohydrates. So first thing that we are going to see is on the basis of the presence of what? Aldehydic or the ketonic group, that is CHO or C double bond O group. So basically, depending upon whether the aldehydic group is free or not, means both the groups, aldehydic, if it's having a free aldehydic group or a ketonic group, there are two types of sugars. Okay, the first one is the reducing and the second one is non-reducing. So reducing and non-reducing sugars means what? There are basically two types of reagents. Okay, that is tolins reagent, and filling solution. Okay, so suppose if a carbohydrate is having a free aldehydic or ketonic group, then it will reduce this filling solution or Tollens reagent. So that type of sugar is called as what? Reducing sugar. So with this, the free aldehydic or ketonic group will react. And hence, what will happen to that? It will reduce. Okay, for example, we can take over here, say, maltose or lactose. In the same way, non-reducing means what will be the opposite? Means because they don't have a free aldehydic or ketonic group, they are not going to reduce that particular solution. So hence they are called as what? Non-reducing sugar. So don't have a free aldehydic or ketonic group. For example, we can take sucrose. Okay, then in the same way, depending upon its characteristics, we have again two types. Okay, that is sugars and non-sugars. So in the name itself, it's there. Now sugar means obviously we know that it will be what? Sweet in taste. Correct? So the basic characteristics is what? If it's a sugar, first thing is it will be sweet. If it's a non-sugar, it will be tasteless. Then the second one is they will be crystalline. Here it will be amorphous. Okay, and the last particular characteristic of sugar and non-sugar is what? Solubility in water. So sugars, because they're crystalline, they will be easily soluble in water. Whereas here, non-sugars will be insoluble. Okay, so we'll be seeing the classification of carbohydrates depending upon how many sugar molecules are attached to them. So in that, basically, we are going to see the three types: monosaccharides, oligosaccharide, and polysaccharide. Okay, so whatever the sugars are there in this category, the first two, that is the monosaccharides and oligosaccharides will come. Whereas in the non-sugar, the last one, which is the polysaccharide. So now what is this? Saccharides means we already know it's sugar. Okay, so mono, oligo, and poly. So mono means single, oligo means 8 to 10 units of the sugar molecules, and poly means what? Many. So more than 10. So this thing we are going to see in detail. So before that, now carbohydrates is a major group molecule. Okay, so it's a macromolecule. So obviously it will be having some kind of linkages present between them. So those linkages over here are called as glycosidic linkage. Okay, in each molecule, the linkages will be different. For example, for carbohydrates, if it's glycosidic, for the protein, it will be peptide. Okay, so it will go on changing according to the macro molecules. So now what is this glycosidic linkage? So glycosidic linkage means nothing but the oxide linkage. Now here it's a dehydration process. So obviously there will be a loss of water molecule. So the oxide linkage will be formed with the loss of the water molecule by the loss of a water molecule. And when does this happen? When two monosaccharides are joined together through oxygen atom. So this particular linkage is called as what? Glycosidic linkage, which we will find in all the carbohydrates. Okay, so depending upon what kind of hydrolysis product we are going to get through this carbohydrates, we have certain classifications. So the first thing, classification of carbohydrates. So this classification we are going to see on the basis of their hydrolysis product what we are going to get. So it is basically of two types. Okay, that is the simple and the complex. So first let's see the simple sugar or simple carbohydrates. So simple means here we'll be studying about the monosaccharides because they are simple sugars. So further being hydrolyzed also you're not going to get any sugar. So this is the final product. So monosaccharides are something which cannot be further hydrolyzed into a simpler component. Hence, they come under the category of what? Simple carbohydrates. Okay, so this particular monosaccharides are further classified into two according to their functional group which is present, which I have already told you, that is the aldoses and ketoses. Okay, that is aldehydic and ketonic group. So the CHO and the C double bond. Okay, so now depending upon how many carbon atoms are present or which type of functional group is present in that particular monosaccharide, there are certain classifications. For example, consider that 3, 4, 5. Okay, so in the first one, we are having the 3 carbon atoms, then 4, and then the 5. So if it's a 3 carbon atom, we'll say it is triose. 4 means tetrose. 5 means pentose. Okay, and it'll go on, hexose, heptose, like that, it'll just go on. In the same way, depending upon whether they are having a aldehydic group or ketonic group. So in front of this particular, this one carbon number term, just you have to put, like if it's an aldehyde, you write it as how? Aldotriose. Suppose if it's a ketone group is present over there, then you will write it as ketotriose. The same is applicable for the rest two also. The tetrose means aldotetrose. Keto means ketotetrose, like that. Okay, so this is how you have to name a particular monosaccharide, depending upon how many carbon atoms are present in them or which functional group is present. Okay, so this is the first one, which is the simple carbohydrates. Second one is what? Complex carbohydrates. So complex means the further two categories we can include, okay, into this, that is the oligosaccharide and polysaccharide. So the complex carbohydrate, okay, which have been divided into two, that is the oligo and the polysaccharide. Okay, so already we know what do you mean by oligo and polysaccharide. So oligosaccharide means what? Whenever that particular carbohydrate gets hydrolyzed, it will give you minimum of 8 to 10 units of monosaccharide units. But if it's a polysaccharide, then what you're going to get is large number or large units, you can say, of monosaccharides. Okay, so depending upon 2, 3, how much you're going to get. For example, if you take the oligosaccharide, again, depending upon how many units you're going to get, different types are there. Okay, like for example, disaccharide. Okay, so minimum we can take over here 2. Okay, so 2 to 10. So over here disaccharide we can take, di means 2. Okay, so C12, H22O11. So in this, we'll be having certain examples. Now here, disaccharide means what? Whenever it's getting hydrolyzed, that particular single thing is going to give you two products. For example, if you take sucrose. So sucrose, when you hydrolyze it, you're going to get glucose and fructose. Same way we can take lactose. So in the lactose, again, made up of glucose and galactose. Okay, then maltose. Here also two components, that is glucose and glucose. So it's a disaccharide. In the same way, you have what? Trisaccharide. So trisaccharide means 3. So for example, raffinose. So when it's hydrolyzed, you're going to get 3 product: glucose, fructose, and galactose. Then again, we can take tetrasaccharide. So tetra means 4. So we can take stachyose. So here also 4 products. So glucose, then it'll give you fructose and 2 molecules of galactose. So this is depending upon how many units it's going to get hydrolyzed and give you the products. In the same way, we can also consider what? The polysaccharide. So polysaccharide means n number of units you'll be having, means many number. So here we can take any, what do you say, the storage components we can take, like for example, starch or cellulose or glycogen. Now all these things are what? The storage components of plants, animals, cell wall and all those stuff. Okay, so here all this are majorly made up of one unit, sorry, one product, that is glucose. So if we take starch, it will be having n number of glucose molecules which are binded together by a glycosidic linkage which forms what? Starch. Okay, so here if you take any of the sugar, like for example, if you take sucrose, sucrose is what? It's a simple, what do you say, table sugar. The same way if you take lactose, it's a milk sugar. Okay, means mostly it's present in milk. Whereas if you take maltose, it's a malt sugar which you get from a barley. Okay, so like that, certain components are there. Even here also, starch, cellulose, glycogen, everything is made up of what? N number of glucose molecules only. The only difference is what? In their structural composition. Okay, so whenever you're going to write, or when you're going to write down that particular formula, okay, either you can write it in open chain form, open chain structure, or in a closed one, or a ring form, you can say. Okay, so in these 2 types, you can write it. So next, again over here, now depending upon, now here I told you, right? Now n number of glucose molecules are there. Okay, so it may be like either a homopolysaccharide or a heteropolysaccharide. So homopolysaccharide means what? Same type will be present. For example, these 3, okay, glucose molecules will be present. So same units throughout the chain. Whereas hetero means what? Different. For example, you can take chitin. Okay, now chitin, which is there, it's the frame, or it's a support for the fungal cells. Okay, mostly for the fungal cells or arthropods, crabs and all those shells which are there, right? So those are made up of what? Chitin. So that particular composition is not only of the single type of sugar, but different types of sugar make up that particular polysaccharide. So this is depending upon whether the same kind of sugar is forming that particular chain or a different kind of sugar. Okay, so you should be able to know the formula, okay, and which type of sugar it is and what products they are going to give when they are hydrolyzed. So moving on forward towards the 2 types of sugars on the basis of where the OH group is present, that is D and L, dextro and rotatory sugars. Okay, so now what is this D sugar and L sugar? So whenever you're writing a particular formula or a structure of the glucose, suppose if you're drawing, so obviously the position of the hydroxyl group, it'll be changing with respect to a particular main group, whether it's the second carbon or the third carbon, it'll change. Okay, so depending upon that only, you have what? D and L. For example, we'll take over here the example of glucose only. Okay, so I'll just CH2OH. And I'll just do a difference over here now in the second thing. Okay. So the first one which is there here, if you see the OH group, the hydroxyl group, it's where it's in the this side, right? The lowest, whatever the chirality center point is there. So over here, it will be what? A D sugar or D glucose. And here it will be L glucose. Okay, so depending upon from the main point where that particular OH group is present. Okay, on that basis, the left and right. D means dextro means right, okay, and the levo means left. So where the OH group is present, depending upon that, you have to write whether it's a D glucose or L glucose or any other component, D glyceraldehyde, L glyceraldehyde, whatever it is. Okay, so basically that particular D is nothing but the right side when the OH group is present, that one, and L is nothing but the left side when it is present. It's called as the L sugar. Okay, so this is what the general structure, just for understanding what is D and L, I have drawn this. Okay, so I told you glucose which is there, it's an important sugar. Okay, because mostly you're going to see this everywhere. Like if you take fruits or if you take any simple table sugar, honey, okay, anything, starch, cellulose, okay, everywhere you will find this sugar. And also in the human blood also, this sugar is a major component of a blood as well, right? So when we come to the characteristics of the glucose, okay, now already we know the terms like whether it's a monosaccharide, poly or oligo, then whether it's having a aldehydic group or ketonic group. Okay, so here we are going to see one more extra thing, that is regarding how the structure of that ring will be represented. For example, we have glucose, we have fructose, right? So both are having same formula almost. Same formula both will be having. But here if I take glucose, okay, glucose characteristic is what? It is a aldohexose. Aldohexose means what? Aldo means it will be having a aldehydic group. Whereas hexose means 6. So 6 carbon compound it is. So usually when it's drawn cyclically, okay, it's a cyclic 6 membered structure. Okay, so that particular structure of the glucose is called as pyranose. It's called as what? The pyranose structure. This is how it comes, 6. Okay, so pyranose structure. So pyran means nothing but it's just basically a heterocyclic component which is having what? Oxygen over here and the 5 carbon ring over here. So this is called as what? A pyranose structure, right? But if it's a fructose, we say it is a furanose structure. Okay, so that fructose I'll be telling you afterwards. So first we'll complete this glucose. So this is what usually glucose are dextrorotatory. So hence you can name it as dextrose as well because of this rotation. Okay, so how this glucose is prepared? So two types you can prepare, by two methods you can prepare this glucose. One is in the labs how you can prepare, and the second one is commercially how the sugar is made, right? So the first thing we are going to see how it's going to prepare preparation of glucose. Two methods. So the first one we are going to see is from sucrose, and this is the basic method. Okay, that is the lab method. And the second one is the by using starch. That's commercial. So in large amount, if you want to produce the glucose, then commercially only you have to opt it for. So the first one is from the sucrose. Okay, so as from the name itself, sucrose is going to give you what? Two components, that is glucose and fructose, right? So the sucrose which is there, that has been boiled over here in the presence of a dilute this one HCl, that is hydrochloric acid, or you can take H2SO4 also. but you have to make sure that both are dilute, not concentrated, dilute. So you have to just boil this sucrose for minimum of 2 hours in the presence of this dilute acid, and the component that you're going to get is what? Sucrose. Okay, so here boiling of sucrose with dilute HCl or H2SO4. Okay, that's for about 2 hours, and then you're going to get is two components. Okay, that is glucose and fructose. But if you're going through a commercial method, obviously you need it in large quantities. So starch is taken over here because n number of glucose molecules it's made up of. So here also almost the same procedure will be there, but the component, because it's starch, so you're going to get much more amount as compared to the, what do you say, the lab method which is there. So over here, starch, what you have to do is you have to again boil the starch, and this also will be in the presence of what? Dilute H2SO4, and then there's a pressure which is given over here, which is 393 Kelvin pressure. And once this is done, okay, you get what? The n number of glucose molecules. And then you have to, whatever the sulfuric acid is present over there, what you have to do is you have to neutralize it. And for doing that neutralization, you have to add into this chalk powder. This is for what? Neutralization. And then what you're going to get is what? The glucose. Okay, so this is how the glucose can be prepared, either in the laboratory on a small scale basis or commercially on a large scale basis. Okay, so in the same way, how the, what do you say, glucose is going to react with different kinds of chemicals, on that basis also you have different reactions. And this is very important because they may ask you, like, if a glucose is reacting with the HI, okay, then what is it going to give? Or when it is reacting with bromine water, what are the products you're going to get? Okay, so that you should know the entire reaction as well as the what products you're going to get accordingly. Okay, so as I told you about the open chain structure, in the same way you will be having a cyclic structure of glucose. Okay, so it's very important to know the cyclic as well as open structure of the glucose as well as the fructose over here. Okay, so the second one which we are going to see is about the fructose. Now, fructose is a fruit sugar, and it's the sweetest. Okay, so there aldohexose it was. Here it is ketohexose. Means 6 carbon, but ketonic group will be present. And here it will be a furanose, 5-membered ring. Okay, so one oxygen atom, instead of 5 carbon, it will be here 4 carbon atoms. Okay, so this is how the structure will be. Okay, so depending here also it will be D-fructo furanose or whatever it is. Okay, depending upon where the OH group is present. Okay, so the almost the formula is same, just the ring will be different over here. So this is all about what? The monosaccharide fructose as well as glucose. So next one we are going to see about the disaccharide and the polysaccharide. So I'll be just simplifying this. Okay, so here we are going to see mainly what is the source and what are the linkages which are present in them. Okay, so I'll just write it down one by one. So carbohydrates. Okay, when it is hydrolyzed, what are the products it's going to give? Then we'll take the linkage, and lastly we'll take whether it's having a reducing property or not. So the first one is the sucrose. Okay, so when it is hydrolyzed, you're going to get glucose and fructose, where the linkage, that is a glycosidic linkage, will be between the C1 and C2. Okay, so C1 of the glucose and C2 of the fructose, and hence it's a non-reducing sugar. In the same way, maltose. So here it's a D-glucose. So here C1 and C4. C1 of one glucose molecule and C4 of the second. So it's a reducing one. Third is the lactose. Okay, so lactose again over here, galactose and glucose. So again C1 and C4. It's a reducing sugar. Then comes the starch. Here n number of glucose molecule is present. Okay, so here now when you come to the starch, it is a little complicated structure because it's made up of amylose and amylopectin. Okay, I'll just tell you what it is. So I'll just complete this table first. So here two components will be there. Depending upon that, the linkages will be present between the carbon number 1 and carbon number 4. So it is amylose and amylopectin. Amylopectin. So for the amylose and amylopectin, over here in the amylose only one C1 and C4 glycosidic linkage will be present. Whereas for the amylopectin, it will be what? C1, C4, and the second one will be C1, C6 linkages. Hence, it's a non-reducing sugar. Then the fifth one is the cellulose. Again, glucose molecule, C1 and C4, non-reducing. Then you have the glycogen. Here also glucose, C1 and C4, non-reducing. Okay, so here if you see the sucrose characteristics, if you see, it's nothing but a cane sugar or a table sugar, as I already told you. So it will be having a glucopyranose structure and a fructofuranose structure, because the components on hydrolysis what we are getting is what? Glucose and fructose. Whereas the linkage is what? C1 of the first, that is the glucose, and the C2 of the fructose. Okay, so what in all the carbohydrates, the linkages which are going to be present is nothing but the glycosidic linkages. Okay, so equal amount of glucose and equal amount of fructose you will be getting over here in the sucrose hydrolysis. And one more important thing is it's also called as invert sugar. The second one is the maltose. Maltose, I already told you, malt sugar, which we get from the barley. Okay, so basically here, whatever it's made up of glucose molecules, only two glucose molecules. So again, C1 of the first glucose and C4 of the second. And hence it's what? A reducing sugar over here. Again, the same, okay, glycosidic linkage only will be present. Same as with the lactose also. Now, lactose is basically what? A milk sugar. So, galactose and glucose is the composition of it. Okay, so it will be having what? A glycosidic linkage between the C1 of the galactose and C4 of the glucose. So, these are all what? The disaccharides. Means whenever it's hydrolyzed, you're going to get two products over here. Whereas, if you see the other one, that is the starch, cellulose and glycogen, all this come under what? A polysaccharide. Okay, so here if I take starch, Okay, starch you will find mostly in the cereals or roots, or okay, you can take potato as an example for the starch. So here, basically, it's made up of two fractions or two compounds, that is amylose and amylopectin. Okay, so amylose and amylopectin both are different depending upon its characteristics. For example, if you take amylose, it is what water soluble. So here this will be insoluble. The percentage which is present in the starch of the amylose is 15 to 20%, whereas over here it's more, that is 80 to 85%. Okay, so because it's water soluble, so it will be an unbranched chain, and this will be the branched one. Okay, whereas if you take the linkage for the amylose and amylopectin, for the amylose, it's what? C1 and C4. And here I already told you two linkages, C1, C4, C1 and C6. Okay, so depending upon, because of the amylopectin only, if you take starch and start dissolving it in the water, it will not dissolve, it just settle down as a powder. Okay, but in the same way, if you're boiling the starch in the water, it will form what? A gel-like component. Okay, so that is because of these two fractions which are present, amylose and amylopectin.